What you'll need to be able to do is tell me what the principles of flow cytometry are in terms of fluidics, optics and detection. And that particular element will be worth around 20 marks, okay? 20 marks. I'll also expect you to know what forward scatter is and side scatter. I will expect you to be able to explain what threshold is, and also be able to tell me why you use different fluorochromes in flow cytometric analysis. You should know the principles of spectral overlap and compensation. And you should also know what is meant by cell gating. Okay, so the next thing you'll need to know is why do we use calibration beads in flow cytometry? And you should have some knowledge of what cellular markers distinguish different T cell subsets. And bringing all this together, you should have some understanding of how this method is used to identify a certain subset of T cells when measuring or monitoring HIV patients. Okay, folks, so before you walk into this practical, you must be able to look down that list of things and be able to answer something on it. If you can't answer any aspect of those questions, I can guarantee you're going to lose marks. Now, what I've done at the end of this particular PowerPoint is provided you with what are the likely questions that you will be asked. Again, just to give you some idea of what you're going to face when you go into the lab. Because the lab work itself, as I said, will probably only last about 45 minutes. And the other 2 hours, 15 minutes are you sitting down, doing calculations, and also answering theory-based questions. Okay? So, just to give a bit of context, so cytometry is the quantification of cell or cellular characteristics. And the spin on this in terms of flow cytometer is it's looking at the quantification of cells or cell characteristics when the cells are suspended in a fluid stream. Okay? So flow cytometry is, as you'll find out, it's a very important method that's used in laboratories, both in terms of research. If any of you are interested in research, you'll probably come across this. But more importantly for biomedical science labs, where one of the main uses of it is for monitoring HIV development and progression. So the flow cytometer, the problem we have here is that in the blood there are a whole range of different blood cells. I think Emer would have went through it with you yesterday. You have a whole range of different leukocytes, for instance monocytes, basophils, eosinophils, neutrophils. And one of the problems we have is we need to have a method that enables us to distinguish between those different types of cells. And the rationale for that is that a lot of these cells are implicated in disease. Therefore, we need a method that's quick, reliable, and reproducible for monitoring these cell populations. And that's where flow cytometry comes in. So it gives us the capacity to measure the amount of different immune cell types that can be important disease diagnosis, and in terms of HIV, more importantly, prognosis. So that's how the disease is progressing and what the risk is of fatality as a result. So flow cytometry provides us with technology for accurate and reliable quantification of a range of different cell types within one patient sample. And the most the best aspect of flow cytometry is that it's high throughput, that is, we can use it in the lab on a day-to-day basis, and that's what enables it to be used in the clinical laboratory. If it was cumbersome and low throughput, it just wouldn't have a place in a biomedical scientist lab. So this is a flow cytometer. These are the basic components. And essentially what it is you take for instance, you take a blood sample, the machine itself takes up the blood, and here you can see the sample. It goes through a nozzle, and this nozzle enables the cells, a mixed population of cells, to actually flow in a single file. And the rationale for getting the cells to flow in a single file is that we can look at one cell individually as it passes through. And when it passes through, it gets to the point where a laser or a light is directed against the cell. The light then gets deflected in all directions, And the deflected light gets detected using these photomultiplier tubes. Okay? And each detector has its own job to do. So for instance here, this will mean nothing to you at this stage, but this detects forward scatter, this detects side scatter, and these ones detect light at different wavelengths. So that's the basic setup of the flow cytometer. On a very basic level, we'll get into more detail as we progress. So the principles of the flow cytometer are fluidics, optics, and electronics. So the fluidics enables a mixture of cells to be directed to flow in a single phase solution, and this process is referred to as hydrodynamic focusing. Okay, so that's a buzzword that you need to be able to incorporate into any answer that you give in your practical report. And of course, this process of hydrodynamic focusing enables cells to flow in a single phase and as a result be analysed individually. The optics consist of lasers to illuminate the particles or cells, or the fluorochromes as we'll come across, and the optical filters will enable different lights of different wavelengths to be directed to certain detectors. So that's the job of the optics, to separate the light that's being deflected by the cell according to wavelength towards a specific detector which will enable us to detect certain markers of a cell. And of course the electronics convert the signal or the light detected into an electronic signal. And that electronic signal then enables us to quantify the number and type of cells present. So, another buzzword that I need you to be aware of is laminar flow. So that idea of having a solution of single cells in single phase, is called laminar flow. Okay? So the blood sample has millions of different cells in it, and what we as scientists want to do is enable the cells to go into a single phase so that we can analyse each cell individually. So that's basically what happens there. Now, this is what happens. The flow cytometer takes up the blood sample, and again it's this huge mixture of cells, and it mixes the blood sample with sheath fluid. And the sheath fluid's added at a certain speed and pressure that pushes the blood sample in towards into a single stream in a manner that it's so thin that only one cell will fit into that stream. Okay? So that process of the sheath fluid being added and causing this effect is called hydrodynamic focusing, and the hydrodynamic focusing causes laminar flow. Okay, this is laminar flow. This process is called hydrodynamic focusing. And this is just an idea, slide to give you an idea of how it works. Okay, so you can see the rate of addition of sheath fluid is 16 to 18 mils per minute, where the sample addition is actually only 0.05 mils per minute. And that creates the different pressure gradients that enables this process of hydrodynamic focusing. Okay, and then this is just another diagram to highlight it again. So what is the sheath fluid? Sheath fluid is phosphate buffered saline, or HEPS buffered saline, and it's basically salty water. It's the easiest way to explain it. And the reason for having the salty water is it creates a nice osmotic balance between the sheath fluid and the cells. What's going to happen if we add water to this instead of sheath fluid? What happens when you add water to a population of cells? Lysis, yeah. I don't know who said it, but osmotic lysis. That's not what we want. So by adding this salty water, or phosphate buffered saline, we prevent osmotic lysis. Okay, so the fluidic system consists of a central core through which the sample is injected, enclosed by an outer sheath fluid. These are pushed together through the system at slightly different pressures, and the sheath fluid's under high pressure and therefore moves faster. As the sheath fluid moves, it creates a drag effect that causes the sample in the central core to narrow. That's just what I explained previously. Okay, so now we've managed to initiate this process of hydrodynamic focusing, and as a result we have laminar flow of a sample. That is a single sample of cells in single vial. So as it moves towards the laser, it reaches the laser, and the point at which it reaches the laser is called again this is another buzzword you need to know, it's called the interrogation point. Okay? So that point where the cell meets the laser is called the interrogation point. And at that point, the laser or light is directed against the cell. And of course, if you direct light against the cell, as soon as it hits the cell, light will get deflected. Okay? And if you have, as we'll come to see later on, if you have a fluorochrome attached to the cell, it will cause fluorescence to be released. But we'll cover that in a little while. What I need you to grasp at this point is you shine a light onto the cell, and as soon as it hits the cell, you get deflection of light. And that leads us on to forward scatter and side scatter. So as you see here, the light source enters the cell, and you're going to get two ways of deflecting the light. One is that all the internal components within the cell, like the cytoplasmic granules, will cause deflection of light out the side. So for instance, if light shines in the cell and hits the nucleus, you'll get light scattered to the side, and this is called side scatter. So side scatter is a reflection of the internal complexity of the cell, and we'll cover that in a little minute. Whereas forward scatter, I suppose the easiest way to describe forward scatter would be like the cell's shadow. Okay? So the bigger the cell, the bigger the shadow. Okay, so light or laser is beamed onto the cell, and the light that goes above and below the cell will cause forward scatter. Okay? And from this, what we get is forward scatter will give us an indication of the size of the cell, which is very useful because some cells are bigger than others. And the side scatter will give us an indication of how complex the internal cell environment is of a cell. And if you have just those two measurements, the side scatter and forward scatter, you can get some indication of what cells you might be dealing with. Okay? So here you go, you shine a light on the cell, and the light that manages to go above and below the cell will give you an indication of the cell size. And the sensor that detects it is the forward scatter sensor. Okay? What I need you to know is forward scatter gives me an indication of the size of the cell. You will definitely have to write that in your report. And likewise, side scatter, and in this case you can see a large cell gives a wide forward scatter, and a small cell will give a narrow forward scatter. So you should get the rationale from that. Now, in terms of forward scatter, this is how it works. So the intensity of the optical signal gets converted to electrical impulse and is recorded by the computer. So here's what happens when you don't have a cell going through the laser. The laser goes through, goes to the detector, and as you can see, you get no signal. But as soon as the cell comes into contact with the laser, light gets deflected and detected, and you get an increase in voltage. And of course, once the cell has passed, the voltage then decreases again. And this is the signal that enables us to tell us how big the cell is. Okay? So that's how the electronic aspect of flow cytometry works. And here you can see, here's an example of side scatter. The more intense the voltage, the higher the voltage, the more complex the cell is. Now this is a very poor slide, unfortunately. But lymphocytes have very little internal complexity. Monocytes have very little, but neutrophils have a lot of cytoplasmic granules, which is why you get a high voltage. Okay? Now, that's how it detects this. This is the results you get at the end. It's a scattergram. Okay? And what you have here, despite the resolution being poor, what you have here is side scatter, and along the bottom you have forward scatter. Okay? And each single spot on that graph represents one cell. Okay? So for instance, if I pick this cell here, no, even better, if I pick this cell here, I know that it has a high side scatter and a high forward scatter. Okay? Whereas if I pick this cell, it has a low forward scatter and a low side scatter. And based on those characteristics alone, I can get an indication of what blood cell it might be. Okay? But from this slide, what I want you to get is each single speckle on this graph represents one cell. So you can imagine the amount of cells that this flow cytometer has measured on that alone. Okay? Again, the resolution's quite poor. It's fine on my computer and on your PowerPoint, but for some reason it's not good on this. So here you have a lymphocyte with very little granules in it, monocytes with very little granules in it, but the neutrophil is filled with granules. So therefore the internal complexity of the neutrophil is very high. So this is what you get. The fact that lymphocytes are A, small, and B, have low internal complexity, that's why I can deduce that this is likely to be lymphocytes. Because they're low forward scatter, which means they're small, and they have low side scatter, which means they have very low internal complexity. Whereas if I move to monocytes, the reason I'm able to distinguish monocytes from neutrophils is monocytes have less internal complexity than neutrophils. So they have high forward scatter, but low side scatter. Okay? Whereas we know neutrophils are A, big, and B, are have a complex internal cytoplasm. So they have high side scatter and high forward scatter. And that's how you can use the very basics of flow cytometer to identify certain cell populations. So based on side scatter and forward scatter, the scientist here has taken the blood sample, put it onto the flow cytometer, and is able to identify certain subpopulations of white blood cells. Does that make sense? Yeah? Okay. Anybody who's having difficulty with this, please look at the recording, have a go at it, and if you find it difficult, come up to me after class and I'll give you a hand. Because you have 2 weeks to get on top of this before the practical. Now, another challenge we have is this scientist has taken the blood sample and he's only interested in lymphocytes, monocytes and neutrophils. But the difficulty is their blood has an incredible, I'm sure you're aware of this, but it has an incredible amount of red blood cells. And red blood cells are just something we don't want, we're not interested in for this function. And if we include red blood cells in the analysis, it'll be very difficult to look at this graph because the red blood cells will overtake the plot. But there is a way to eliminate red blood cells from the analysis, and one way we do this is through a concept known as threshold. Now, if you can imagine, red blood cells are a lot smaller than leukocytes, and they don't have much complexity. So red blood cells are going to appear here, okay? Because they are small, so they're going to have a small forward scatter. They're not complex internally, so they're going to have a low side scatter as well. And what this scientist has done, has told the computer not to measure anything in this range. I'm sure you can see it from here to here, the whole way up, there's nothing being recorded. That's not accidental. The scientist has told the software, eliminate anything that is as small as this and as with low complexity. Okay, does that make sense? And that's the concept of threshold. So when you're asked about threshold, you'll write that it's a way to eliminate cellular events that you are not interested in. Okay? So it's elimination of small debris, such as lyse red blood cells or lyse cells, but also red blood cells, intact red blood cells as well. So the detector is instructed to only detect events that are above a particular threshold. And threshold is usually determined by forward scatter. That is, it's usually determined by the size of the cell or particle. And this enables the user or the scientist to eliminate noise or debris. But the one thing we need to be careful is that you don't set the threshold too high. If you set the threshold too high, you might miss events that you're actually interested in. Okay? And here's an example here. Again, the resolution isn't great, but you can see here the user has set a threshold at 200 forward scatter. And you can see very thin line that doesn't record anything. But here you can see some cells are being detected, or some events are being detected. So the threshold of 200 just isn't good enough. So then he sets it, or the scientist sets it at 5,000. And here you can see there's still some cellular debris. So then they progress and set the threshold to 40,000. And now we're in the perfect zone because there's no events that we're not interested in. Okay, so that's how you would set your threshold. It's almost like a suck it and see approach where you try different thresholds until you identify a threshold that will work for you. Okay? Okay. Now I'm just going to play a short video to give a brief overview of everything I've talked about so far. Okay, folks, appreciate silence during this, please.
In this animation, we will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. the computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules.
We'll cover that in a little second. Okay, so that's the basics of the flow cytometer up until this point. So, we're now at the point where we can identify certain cells based on their size and their internal complexity, but that can only bring us so far. The difficulty we have is we're going to encounter cells which are important in disease that are actually of a similar size to other cells. And the difficulty we have is we want to be able to distinguish the cells that are causing disease from other cells which are relatively normal. An example of that would be T cells. Okay? There are a range of different T cell subsets, which Emer might have covered, but certainly I'll be covering it in week 3. This brings along a problem. If all the T cells are of similar size and complexity, how do we tell the difference between the different subsets? We certainly can't do it with forward scatter and side scatter because they have the same physical characteristics. So, forward scatter versus side scatter can only provide so much. And as I said, the example would be taking a range of different T lymphocytes that have different functions but actually physically are very similar. The type of cell subsets that you're going to know about are CD4 cells. So CD4 cells are helper cells. They help, they're cells that help initiate the adaptive immune response. But of CD4 T cells, there are different subsets within it. There's a subset called Th1, which can help with the response against virus. There's a subset called Th2, which can help in a response against bacteria or parasite. And you have Th17, CD4 cells, which have another role at mucosal surfaces, but are also implicated in autoimmune diseases. And secondary to that, you also have CD8 cells. And CD8 cells are cells that, generally speaking, are directed against viral infections or against tumour cells. Now, the difficulty is, as you can see from the picture, they all look the same. So physically, they're all the same, and we need to come up with a way to differentiate each cell from the other if flow cytometry is going to work for this function. So how can we differentiate these cells? We basically differentiate it based on the receptors that each of these cells express on their surface. And similar to what Emer covered with the ELISA, what we're going to do is develop antibodies that are going to bind the specific receptors on the cell. Okay? And I'm going to take you through what receptors are important for this particular practical. So the first thing to note is that all leukocytes have CD45 expressed on their surface. Okay? This little yellow circle. So you need to be able to convey that CD45 will enable us to identify leukocytes. But of course all leukocytes have similar characteristics. So the next thing we want to do is identify, well, what receptor expressed on lymphocytes makes them different from a monocyte or different from a neutrophil. And the receptor or the molecule expressed on the surface of lymphocytes that makes them distinguishable from other leukocytes is CD3. Okay, so CD45, CD3, and then the next thing we want to know is what makes CD4 cells different from CD8. And of course, as simple as that, it's the CD4 molecule and the CD8 molecule. So all we have to do in terms of applying that to the flow cytometer, what we then have to do is develop antibodies that will bind to CD45, antibodies that will bind to CD3, and antibodies that will bind to either CD4 and CD8. Okay? And if we're able to do that, we will be able to use the flow cytometer to distinguish each cell population. And I'll take you through how that's possible. So again, to identify leukocytes, CD45, to identify T lymphocytes, CD3, and to identify CD4 lymphocytes from CD8, we use the CD4 and CD8 receptors. Now, because we want to look at 3 or 4 different receptors at the same time, we then have a new challenge of, well, how do we distinguish between these different receptors if they're being detected by the flow cytometer all at the same time? It's quite simple. We use these molecules called fluorochromes. And fluorochromes are basically like adding a colour to the antibody. So for instance, I'll add a red colour to the CD45 antibody, I'll add a green colour to the CD3, I'll add yellow to CD4, and I'll add purple to CD8. So that when those cells go through the flow cytometer, when they pick up those colours, they know that the cell has CD45, CD3, and so on. So we create antibodies that specifically bind to the cell markers that we're interested in. But we need to identify fluorochromes to attach to the antibodies to enable us to distinguish between the different antibodies attached to the cell. Each cell marker requires a different fluorochrome to be used. Okay? And when excited by the laser, the fluorochromes will light up and give off a signal. And this light given off when the fluorochrome is excited will be given off at a specific wavelength and detected by a specific detector. So this is basically what happened. You have your blood sample and you start to label the cells that you want. So the first thing we do is we add an antibody to CD45. Okay? Now here we have a CD8 T lymphocyte and a neutrophil. And at this point, it's quite clear for this practical, we have no interest in neutrophils. Okay? But the first thing we want to do is isolate leukocytes in general. So we add an antibody with a green fluorochrome to bind to CD45. At this stage, we know we have leukocytes. The next thing we want to do is add an antibody that will enable us to distinguish between a CD8 and a neutrophil. So we add an antibody which will bind to CD3. And at this point, we've added a red fluorochrome so that we can distinguish between these two antibodies. And as you can see, neutrophils don't have CD3. So that's a good thing. We can now distinguish between what's a neutrophil and what's a lymphocyte. The next thing we want to be able to do is distinguish between neutrophils and lymphocytes again. So we'll add a fluorochrome we add an antibody that will bind to a receptor that's unique to neutrophils, GR1. Okay, and again, it's a different colour fluorochrome. But the most critical aspect at this point is to identify that it's a CD8 cell. And by doing that, we add an antibody with a yellow fluorochrome that will enable us to identify the CD8 receptor. Okay? And that's how this happens. All you simply do is take the blood sample, add a cocktail of antibodies, So all these antibodies will be added at the same time, and only the cells with the receptors will attach the antibodies. The excess antibodies, similar to ELISA, the excess antibodies will be washed off. Okay? And that's how we use flow cytometry to identify cells of similar size and internal complexity. Okay? So this is what you get, okay? So now we have information on what cells have CD45, what cells have CD3, what cells have CD8 and CD4. Now we're at the point where we have all this information and we just have to pick out the bits that we like. So here we have side scatter and CD45, okay? So this axis is detecting the fluorochrome that's associated with CD45. So these cells have high CD45, therefore they're the cells that we're interested in. And what we simply do is we say we're not interested in these cells, we're only interested in cells with high CD45. Because of course the cells with high CD45 are leukocytes. So we gate this region, okay? And by gating, what we're doing is selecting all of these cells and moving them to this histogram, okay? So this histogram has all of these cells, but none of these, okay? Now that we've moved it to that histogram, we now have GR1, which is a neutrophil marker, against CD3, okay? So any cell that has high CD3 and low GR1 is a lymphocyte, okay? So we gate this region because it has low GR1 and high CD3. And we tell the computer to take all of these cells and move them to this histogram. So all of these cells are now plotted here, and here you have CD4 against CD8. And the way we identify CD8 is that it has high CD8 but low CD4. And the way we identify CD4 is it has low CD8 and high CD4. And that's the process you go through to identify cell populations. Okay? So the other thing just to remind you is that each single speckle on these graphs equates to one cell. And the software can calculate how many cells are in each area. Okay? So it's just a stepwise progression. Okay? Once again, here we are, side scatter and the fluorochrome for CD45. So we go, well, we want cells that have high CD45. So we go straight to the high CD45 and we arbitrarily pick this region. Okay, take all that information and put it into a new plot where you have a fluorochrome for the neutrophil marker GR1 against the fluorochrome for CD3. At that point we know we want cells that are high in CD3, so we go across, okay, this is high, and we pick a region of high CD3 with relatively low GR1. We take these cells across to this plot where you have a fluorochrome for CD4 and a fluorochrome for CD8. And by that way we're able to identify high CD8 and high CD4. And that's the process. Okay? So, you need to be familiar with the process of gating, which is what I've just talked you to. Gating is a process of selecting cell populations of interest and eliminating cells not of interest. Okay? Great. Now, lasers and fluorochromes. So the thing is, we've attached different fluorochromes to the antibodies, but we need a laser to stimulate the fluorochrome to give off light. So fluorochromes are needed to excite, or fluorochromes are excited by the light or the laser. When they're stimulated by the laser, they emit light at a given wavelength, giving a specific colour. The use of different fluorochromes emitting different colours enables us to measure different cell markers. And with a range of different cell markers that we identify, we can try to isolate different cell populations. So, another thing to consider is what we have here is a range of different fluorochromes, okay? All these fluorochromes here can be used for flow cytometric analysis. But the other additional factor is you can use different lasers, okay? So for the blue laser, I can use these range of fluorochromes. For the red laser, I can use these fluorochromes. For the violet laser, I can use these fluorochromes. And the reason I'm highlighting this is that the increased number of lasers that a flow cytometer has enables the flow cytometer to use a greater range of fluorochromes. And a greater range of fluorochromes you can use means there's more cell markers that you can look at. Okay, there might be some cells, particularly in research, there might be some cells where you might need 7 or 8 cellular markers to identify it. And the difficulty is if you have 7 or 8 cellular markers that you need to identify, that means you need 7 or 8 different colours to enable it to identify each one of those. And if you have increased number of lasers, you have the capacity to increase the number of fluorochromes you can use. Okay? And again, this is just another example of a cancer centre that uses different lasers to identify different uses different lasers to enable it to use a greater range of fluorochromes, which enables it to identify a range of different markers. Now, this is an example of the spectral capacity of different fluorochromes. Okay? So if you stimulate the fluorochrome at 488, this is a 488 laser, you stimulate it at 488 and it causes, for instance, this BD Horizon BB515 will give off excitation at this range here. Whereas this fluorochrome, if you stimulate at 488, it will give off fluorescence at this wavelength. Now, the reason I put this up is A, to show you the range of different fluorochromes that a specific laser can use, but also to show you the difficulty in that how is the flow cytometer going to differentiate this fluorochrome from this fluorochrome, or this fluorochrome from this fluorochrome. And it's going to lead me on to talk about this issue of spectral overlap. Okay, now you can see, for instance, with this, it will be quite easy to distinguish the signal for this fluorochrome and this fluorochrome, because there is no overlap. They're actually quite separate. Now, what I'm going to talk about is, if you design your experiment properly, you can use a lot of these fluorochromes, but you just have to adjust for the overlap. Okay? And then here's an example of a different laser that can use different fluorochromes. As you can see here, these fluorochromes are more spread out than the previous one. So each laser will be specific to a particular set of fluorochromes. So as I said before, what's the point of a flow cytometer having a greater range of lasers? Basically that you have the capacity to use a greater range of fluorochromes, and therefore look at a greater range of cellular markers. So, you've got all these fluorochromes giving off light at different wavelengths. How do we separate these different lights? We use optical filters. Okay, and these are the different type of optical filters that a flow cytometer can use. It can use a long pass filter. So if you have a long pass filter that is a 560 long pass filter, essentially what it does is will deflect light that's higher than 560. Okay? If you have a short pass filter, it will allow it to deflect light that is below 550. And if you have a band pass filter, it enables the flow cytometer to select a light wavelength of a specific range. Okay? So between 530 and 525, for instance. Okay? And using this range of filters enables the flow cytometer to siphon off, if you like, the signal for each of the different fluorochromes. And by siphoning off each of the signals, each of the wavelengths, it enables separate detectors to quantify each of the fluorochrome signals. So here you are an example. So you have excitation laser. It hits the cell, and each of these filters will enable the wavelength of the specific fluorochrome to be detected. Okay? So for blue, here, it will detect light that's less than 500. For the green, it will allow it to detect light that's less than 540. Yellow and orange, 590. And then the remainder will go through to the red filter. So you can see each of these detectors then will send a signal to the digitizer. And at that point, I'll be able to tell how many cells, or did the cell have a high amount of yellow fluorochrome. And if it had a high amount of yellow fluorochrome, I know what type of receptors that it expressed. Okay? Now, that's the basic process. Forward scatter, side scatter, tells us the size of the cell, the internal complexity. Then we use antibodies with attached fluorochromes to identify specific cell markers that are unique to a cell population. And then we use optics to siphon off the fluorochromes at different wavelengths, the light emitted by the fluorochromes at different wavelengths, to detect the different fluorochromes. Now we're moving on to the difficulty of the clash of different fluorochromes. As I said before, certain fluorochromes will be difficult to distinguish. The detector will find it difficult to distinguish between certain fluorochromes. And this concept is known as spectral overlap. So you'll be asked a question on spectral overlap. Okay? Spectral overlap and compensation. So spectral overlap is when small amounts of light from one fluorochrome is picked up by two detectors close in wavelength. So basically what it is one fluorochrome is detected by the wrong detector, giving a false positive. Okay, so this spectral overflow can give misleading results. But there is a way around it. If we know how much overlap there is between one fluorochrome and another, we can tell the machine mathematically how to compensate for that. Okay? So the software can use a mathematical correction process that enables us to address this spillover. So if you have the signal for CD45, if you have part of that signal spilling over into the signal for CD3, then if you know how much of that spillover occurs, you can compensate to avoid a false signal. Okay? So compensation is having awareness of the spectral overflow between two fluorochromes and instructing the computer to compensate for that mathematically. Okay? It is a difficult concept to grasp. My advice is take the notes and the recording, try to get a grasp of it. If you're finding it difficult, if you're finding it a difficult process, I'm more than happy to go through it again. Okay, and this is a sort of visualization of how this can happen. So here you have a detector for fluorochrome A, and here you have a detector for fluorochrome B. Now, fluorochrome 1, detector for fluorochrome A is detecting a huge amount of the signal for fluorochrome A. That's great. But you see here it's detecting a little bit of signal for fluorochrome B. Now the problem with this is this detector will add the signal from this on top of A, giving a false positive, i.e. giving it a higher reading than it really should. And likewise, the detector for fluorochrome B will add this signal from fluorochrome A on top of the signal for fluorochrome B, again giving a false high result. So essentially what it does is compensation enables us to take this away from the overall signal, giving us the true measurement here. And likewise with B. And that process is called compensation. So compensation is required to address the issue of spectral overlap between two different fluorochromes. Okay? Now, why would you ever choose to do this? The reason you choose to do this, one could propose that you just pick two fluorochromes that don't have spectral overlap. But if we were to go down that route, it would limit the amount of fluorochromes we can use. By using this process of compensation, it enables us to use a wider range of fluorochromes and just undertake this mathematical equation to adjust. Okay? Now, if I was to do an experiment where I had a cell population where I only wanted to look at two cell markers, I would develop I would add fluorochromes to antibodies that were completely different colour, because it just makes the job easier. For instance, if I selected this fluorochrome and this fluorochrome, then you know there's not going to be any spectral overlap. But that's an ideal scenario. Generally, when you're doing this type of analysis, you're going to need 3 or 4 different cell markers. Now, it's going to be very difficult to pick 3 different fluorochromes from that graph that don't overlap on some way. So that's the rationale for using spectral overlap. It's just a necessity if we want to look at a wider range of markers on cells. So, compensation. Compensation is required for the flow cytometry experiment just based on the physics of fluorescence. Using different fluorochromes to measure different cellular markers enables specific cell types to be identified. Each fluorochrome can be excited at 488 if you're using a 488 laser. It can be excited at 488, however, the emission detection of each fluorochrome is different, is emitted at different wavelengths. And that's the concept. The laser stimulates the fluorochrome and it emits a wavelength. And the fact that different fluorochromes emit light at different wavelengths enables us to distinguish between them. As I said, the problem we have is the emission detection of one fluorochrome may spill over into the detector of another fluorochrome, giving false positives. And compensation is a mathematical correction that can be used to address this spillover. Compensation enables multiple antibodies bound with fluorochromes to target a wider range of markers on cells. And I've just put in a little link there that will maybe give you more detailed insight into compensation. Okay, let's see if we can get this going.
Flow cytometry is a powerful technique for the analysis of multiple parameters of individual cells within heterogeneous populations. Flow cytometers are used in a range of applications, from immunophenotyping to ploidy analysis to cell counting and GFP expression analysis. The flow cytometer performs this analysis by passing thousands of cells per second through a laser beam and capturing the light that emerges from each cell as it passes through. The data gathered can be analyzed statistically by flow cytometry software to report cellular characteristics such as size, complexity, phenotype, and health. In this tutorial, we will look at how a flow cytometer works, how scattered light and fluorescence are detected by a flow cytometer, and how the resulting data can be analyzed. This view shows the primary systems of the flow cytometer schematically. These are the fluidic system, which presents samples to the interrogation point and takes away the waste. The lasers, which are the light source for scatter and fluorescence. The optics, which gather and direct the light. The detectors, which receive the light. And the electronics and the peripheral computer system, which convert the signals from the detectors into digital data and perform the necessary analyses. The interrogation point is the heart of the system. This is where the laser and the sample intersect, and the optics collect the resulting scatter and fluorescence. First, let's talk about how the sample is delivered to the laser. Here we see how the sample is transported through the interrogation point. For accurate data collection, it is important that particles or cells are passed through the laser beam one at a time. Most flow cytometers accomplish this by injecting the sample stream containing the cells into a flowing stream of sheet fluid or saline solution. As you can see, the sample stream becomes compressed to roughly 1 cell in diameter. This is called hydrodynamic focusing. In fact, flow cytometers can accommodate cells that span roughly 3 orders of magnitude in size. In most cases, cytometers will be detecting cells between 1 and 15 microns in diameter, although through the use of specialized systems, it is possible to detect particles outside this range. Now let's see how laser light is used to detect individual cells in the stream. As a cell passes through the laser, it will refract or scatter light at all angles. Forward scatter, or low-angle light scatter, is the amount of light that's scattered in the forward direction as laser light strikes the cell. The magnitude of forward scatter is roughly proportional to the size of the cell, and this data can be used to quantify that parameter. But how can we record this scattered light? Light is quantified by a detector that converts intensity into voltage. In most cytometers, a blocking bar, called an obscuration bar, is placed in front of the forward scatter detector. The obscuration bar prevents any of the intense laser light from reaching the detector. As a cell crosses the laser, light is scattered around the obscuration bar and is collected by the detector. The scattered light received by the detector is translated into a voltage pulse. Because small cells produce a small amount of forward scatter, and large cells produce a large amount of forward scatter, the magnitude of the voltage pulse recorded for each cell is proportional to the cell size. If we plot a histogram of these data, smaller cells appear toward the left, and larger cells appear toward the right. A histogram of forward scatter data is a graphical representation of the size distribution within the population. But such a graph only presents one-dimensional data. Next, let's look at side scatter. As we have already seen, a cell traveling through the laser beam will scatter light at all angles. Light scattering at larger angles, for example to the side, is caused by granularity and structural complexity inside the cell. This side-scattered light is focused through a lens system and is collected by a separate detector, usually located 90 degrees from the laser's path. The signals collected by the side-scatter detector can be plotted on one-dimensional histograms, like we saw for forward scatter. The one-dimensional histograms we have seen so far do not necessarily show the complexity of the cell populations. For example, what appears to be a single population in the forward scatter histogram is, in reality, multiple populations that can only be discerned by looking at the data in a second dimension. This is done through the use of two-dimensional dot or scatter plots. You can see that the peaks from the forward and side scatter histograms correlate with the colored dots in the scatter plot. Now we can view the results obtained when we create a scatter plot using forward and side scatter data from a typical peripheral blood cell run. The populations that emerge include lymphocytes, which are small cells possessing low internal complexity, monocytes, which are medium-sized cells with slightly more internal complexity, and neutrophils and other granulocytes, which are large cells that have a lot of internal complexity. This multi-parametric analysis is the real power of flow cytometry. Now let's take a look at another parameter that can tell us more about cell structure and function: fluorescence. As a brief review, fluorescence is a term used to describe the excitation of a fluorophore to a higher energy level, followed by the return of that fluorophore to its ground state with the emission of light. The energy in the emitted light is dependent on the energy level to which the fluorophore is excited, and that light has a specific wavelength and consequently a specific color. One of the most common ways to study cellular characteristics using flow cytometry involves the use of fluorescent molecules, such as fluorophore-labeled antibodies. In these experiments, the labeled antibody is added to the cell sample. The antibody then binds to a specific molecule on the cell surface or inside the cell. Finally, when laser light of the right wavelength strikes the fluorophore, a fluorescent signal is emitted and detected by the flow cytometer. How is this fluorescence information collected? The fluorescent light, coming from labeled cells as they pass through the laser, travels along the same path as the side scatter signal. As the light travels along this path, it is directed through a series of filters and mirrors so that particular wavelength ranges are delivered to the appropriate detectors. Fluorescence data is collected in generally the same way as forward and side scatter data. In a population of labeled cells, some will be brighter than others. As each cell crosses the path of the laser, a fluorescence signal is generated. The fluorescent light is then directed to the appropriate detector, where it is translated into a voltage pulse, proportional to the amount of fluorescence emitted. All of the voltage pulses are recorded and can be presented graphically. What if we want to do a two-color experiment? We need to look at the spectra of the two fluorophores to see if they are compatible. Alexa Fluor 488 and phycoerythrin, or RPE, are commonly used together. For these two fluorophores, 488 nanometer light is an efficient excitation source. When excited with 488 nanometer light, you can see that the emission peaks for these two dyes are far enough apart so that discrete emission data can be collected. Compatible dyes such as these allow scientists to easily detect two colors from a single laser. If we analyze data from the two-color experiment using a scatter plot, four distinct populations emerge. Looking at the dot plot in terms of quadrants, cells with only bright orange fluorescence appear in the upper left quadrant. Cells with only green fluorescence appear in the lower right quadrant. Cells with both bright green and bright orange fluorescence appear in the upper right quadrant. And finally, cells with both low green and low orange fluorescence appear in the lower left quadrant. Multiple fluorescence parameters are necessary to dissect complex biological systems. How does the flow cytometer collect discrete fluorescence data for the Alexa Fluor 488 and RPE fluorophores? Filters are available that can capture the peak fluorescence from each of these molecules. A 530 nanometer bandpass filter will collect most of the Alexa Fluor 488 peak, and a 585 nanometer bandpass filter will collect the bulk of the RPE peak. Using these filters, the proportional amounts of Alexa Fluor 488 and RPE fluorescence can be recorded for each cell. You can see that portions of each emission peak overlap one another. This is called spectral overlap, and we will talk about how to deal with this in the next tutorial. Let's take a moment to talk about filter nomenclature. Filters are normally defined by one of two parameters, either the center point for a bandpass filter or the cutoff point for a long or short pass filter. This is a typical bandpass filter specific for RPE. It has a center point of 585 nanometers and a width of 42 nanometers. So this filter optimally passes light in the wavelength range of 564 to 606 nanometers, which corresponds to the emission peak of RPE. Other filters used to resolve this peak include a 550 nanometer long pass filter and a 610 nanometer short pass filter. One final important point regarding data collection is the use of a threshold. If every single particle passing through the laser caused the instrument to collect data, the data pool would be dominated by information coming from a very large number of minute particles, like platelets and debris. To prevent this, a threshold is set such that a certain forward scatter pulse size must be exceeded for the instrument to collect the data. On the histogram, the blank area represents the small cells and debris that are excluded from analysis by the threshold. This means that the majority of events that the cytometer collects are the cells of interest. It is important to realize that the small particles are still passing through the instrument. They are just being ignored. We've come to the end of this tutorial. Our journey began with a look at the systems that make up the flow cytometer and how those systems work together to collect information on cells as they pass through a laser. We went on to examine in more detail how cytometers detect light scatter and fluorescence, and how that information can be viewed on various plots. In summary, flow cytometry is a unique tool, providing scientists with a way to gather statistical data on large numbers of cells and use that information to correlate multiple parameters within a cell population. 4 to 6 color experiments are becoming easier. A few labs in the world are able to distinguish up to 18 colors simultaneously. In the next tutorial, we will talk in more detail about some of the trickier aspects of data analysis that are employed in flow cytometry, especially when 2 or more fluorescent colors are used.
Okay, so that's mainly for the purposes of revision. Should give you an overview. Okay, so next issue is, and this will form part of the main question in the experiment, is when you put a blood sample onto a flow cytometer, you don't actually know how much volume of blood is being used. And this is a difficult scenario because diagnosis of certain diseases will be that a certain number of cells per cubic millimeter of blood will indicate disease. So the challenge we have is in order to get this information, we need to know exactly how many cells were detected per cubic millimeter of blood. And in the flow cytometer standard flow cytometer, there's no actual way to measure the volume used. And the way we do this is we use fluorescent beads. So if we spike the blood with fluorescent beads, the amount of beads that are detected by the flow cytometer will indicate the volume of blood that has been used for that particular analysis. Okay, and these are the fluorescent beads. The concept of the fluorescent beads is that they will have extremely high fluorescence on one fluorochrome and on another. And the reason you want them to have extremely high fluorescence, these beads, is so that they're absolutely separate from the rest of the blood sample. Okay, you see the population here. These are little synthetic beads with fluorochromes attached to it. You then put that into the blood, and the amount of these beads that you detect will give you an indication of the volume of blood that was used for that analysis. So the concept could be that you put 1 million beads into 1 ml of blood, you then put it onto the flow cytometer, and if you detect 500,000 of these beads, how much sample did the flow cytometer use? So you've just put 1 million beads into 1 ml of blood, and then the flow cytometer was able to detect 500,000 of these beads. What volume do you think the flow cytometer used? Someone said what? Yeah, exactly. So you get the concept that if you know how many beads are in the blood, then the amount of beads detected by the flow cytometer should enable you to tell how much of that blood was used for the analysis. Okay? So when you go to do your calculations, this is the main bit you'll need to get a hold of, the volume used. Because the prognostic value for CD4 cells in HIV is dictated by the amount of cells per cubic millimeter. So in order to assess the patient against the recommendations, you need to work out the volume of blood that you measured. Okay, the other thing that the flow cytometer can do is actually sort cells can separate individual cells from the mixture so that you can then use them for further analysis. So this would mainly be used for research purposes. Basically what happens is you tell the computer that if you detect, for instance, green fluorochrome, which would be indicative of, say, CD8, you tell the computer to send a signal to an electric wire, and as soon as that cell passes past the wire, the computer sends a little shock to the cell. And when it sends the shock to the cell that's been detected, it creates a charge, and then the magnet of the flow cytometer will enable that cell to be siphoned off from the waste. Okay? So you detect the cell that you want, the computer senses that the cell's there, it then sends an electrical charge to the cell that's passed through, and by applying a charge to the cell, it creates the cell itself develops a charge. If the cell develops a charge, you can get it to siphon off into a separate container from the waste. And in that way, you can actually collect cells for research purposes. Okay, so what questions will you be assessed on? You'll be assessed on the overview of how the flow works. You'll be asked to explain and provide an understanding of different fluorochromes, spectral overlap, etc., etc. Okay? Now, I think we're going on a bit at this stage. There is other information on what HIV involves, but I'll try to provide a recording for that. Okay, folks? Thank you. Okay, so now a background to HIV and AIDS, which will be critical to the practical itself. So CD4 helper cells, or CD4 T lymphocytes, are helper cells, and the thing you need to be aware of is that CD4 lymphocytes are critical to any adaptive immune response. So if you want an immune response against a virus, you need CD4 helper T cells to provide help to CD8 cells. If you want an immune response against a bacteria or a parasite, you need CD4 helper T cells to provide help to B cells. So CD4 T lymphocytes are critical to any immune response. And damage to CD4 cells or to CD4's T cell populations means the body's unable to produce an adequate immune response, and as a result the body will become immunodeficient and susceptible to infections. So the HIV infection life cycle is detailed here. As you see, HIV will bind to the CD4 receptor and CCR5 receptor of CD4 cells to gain entry. So the HIV virus then uses its cellular machinery to replicate, and once it replicates, it then subsequently goes on to infect more CD4 helper cells. CD4 cell populations are depleted as they'll die from apoptosis as a result of CD8 T cell killing these infected cells. Because, of course, CD8 or cytotoxic T cells are the cells that help kill virally infected cells, and because the CD4 is infected with a virus, the CD8 cell will kill it. So, what you then have is a decrease in CD4 cells, and because CD4 cells help recruit immune cells and are critical to any immune response, you get you get a reduced risk of infection. But monitoring absolute CD4 populations as well as CD4-CD8 ratio helps monitor disease progression in HIV. It also helps you monitor response to treatment. And for the purposes of this practical, the monitoring of CD4 and CD8 cell populations is monitored through flow cytometry. So HIV is commonly diagnosed by testing your blood or saliva for the presence of antibodies to the virus. Unfortunately, these types of HIV tests aren't accurate immediately after infection because it takes your body some time to produce the antibodies. It can take up to 12 weeks. In rare cases, it can take up to 6 months for a HIV antibody test to become positive. A newer type of test checks for HIV antigen. That's a protein produced by the virus immediately after infection. This test can confirm a diagnosis within days of infection. An earlier diagnosis may prompt people to take extra precaution to prevent transmission of the virus to others. There's also increased evidence that early treatment may be of benefit. So test to tailor your treatment. If you're diagnosed with HIV or AIDS, several types of tests can help your doctor determine what stage of the disease you're at. One of these tests are a CD4 count, because of course HIV targets CD4 helper T cells and essentially result in a decrease in CD4 T cells. Therefore, if you measure the amount of these cells present in the blood, you may get some indication of disease progression. So CD4 cells are a type of white blood cell that's specifically targeted by HIV. A healthy person's CD4 count can vary from 500 to 1,000. Even if a person has no symptoms, HIV infection progresses to AIDS when his or her CD4 cell count goes below 200. And viral load's an important thing to consider. This test measures the amount of virus in your blood, and studies have shown that people with higher viral loads generally fare more poorly than those with a lower viral load. And you can also have drug resistance. This test determines whether the strain of HIV you have will be resistant to specific HIV medications. And might be able to also determine which ones are most likely to work better. So CD4 to CD8 ratio cell counts. The flow cytometer is an important tool in monitoring the decline of CD4 cells. A normal CD4 cell count would range from, say before, 500 to 1500 cells per cubic millimetre. And a normal CD4 to CD8 ratio is usually 2 to 1, but in patients with AIDS, this can change to 1 to 2, and that's a result of decline in CD4 helper T cells. So loss of CD4 cells results in loss of HIV-specific immune response, recall antibody response, and finally non-specific immune response in the AIDS stage. This leaves the body extremely susceptible to infections that in general in normal would be able to handle. So the fact that the HIV is infecting CD4 cells and the CD4 cells are being killed off means that memory that we have for general infections that we would normally be protected from, the AIDS sufferers at increased risk. So the assessment questions are listed here, and these are the questions that you're likely to be asked in the assessment. I would strongly advise that you get up to scratch with the role of CD4 in AIDS, and also read the literature and the supportive material about the role of CD4 cells in AIDS. Okay folks, thank you.